Cas9 is a protein that can cut both strands of DNA, but the protein does not choose its destination alone. An RNA guide provides the address. This 2012 paper asked a practical engineering question. Could the natural two-part RNA guide be redesigned as one molecule without losing precise DNA cutting? Start with the physical setup. Cas9 is the cutting protein. The CR-RNA contains letters that match a chosen DNA sequence. The tracker RNA forms a scaffold that Cas9 recognizes. And the DNA needs a nearby PEM, a short signal that helps license target recognition and cleavage. In the natural type 2 CRISPR system, the CR-RNA and tracker RNA are separate molecules. They base pair with each other, forming one functional guide complex. The CR-RNA supplies the changing target match, while the tracker RNA supplies a more reusable structure that fits Cas9. The authors connected the end of the CR-RNA to the beginning of the tracker RNA with a small hairpin loop. The front of this chimera still matches the DNA. The back still forms the Cas9 binding scaffold. Two biological jobs are now encoded in one continuous RNA transcript. Figure 5 compares two versions of this engineered guide. Both aim at the same DNA target, but they retain different amounts of the tracker RNA structure. The longer chimera guides efficient plasmid cleavage. The shorter version works poorly, showing that extra scaffold nucleotides are important for binding or target recognition. How is cutting measured? The target sequence sits inside a circular plasmid. After the Cas9 reaction, a second restriction enzyme cuts at a known site. If Cas9 also cut in the predicted place, the plasmid becomes fragments of predicted lengths. Gel bands reveal those physical fragment sizes. The authors also used a short, labeled double-stranded DNA target. This makes the cleavage product easy to measure precisely. The working single guide and a natural dual RNA produce the same sized cut product. So fusing the guide changes the packaging of the RNA, not the position where Cas9 cuts. A single successful target could still be a special case. The stronger test changes only the target matching section. The researchers designed 5 chimeric guides against 5 sequences in the green fluorescent protein gene. All 5 produce the expected plasmid fragments while reusing the same overall scaffold architecture. The direct observation is modest and concrete. Purified Cas9, guide RNA, and target DNA produce fragments of the expected sizes in vitro. From that, the authors infer sequence-specific programmable cleavage. This is strong molecular evidence, but it is not yet evidence about delivery, safety, or outcomes in a patient. The programming rule is simple. Choose a DNA target with a compatible nearby PEM. Rewrite the guide's roughly 20-letter matching section so it complements that target. Keep the Cas9 binding scaffold largely fixed. The RNA sequence changes the address, while the protein and structural interface can be reused. This paper established the core mechanism in purified reactions and proposed genome editing potential. It did not by itself solve how to deliver Cas9 into the right cells, control unintended cuts, or prove clinical benefit. Those are separate engineering and biological questions that require their own experiments. The central insight is a clean separation between address and tool. Cas9 supplies the cutting chemistry. The RNA scaffold connects to Cas9. A short, replaceable RNA sequence specifies the DNA address. By fusing the natural 2-RNA guide into one programmable molecule, the paper created an unusually simple way to retarget the same cutting protein.